Hi everyone, welcome to lesson 3.7 on the S phase, also known as DNA replication. So we're going to start with a little bit of a review for DNA. So first we have DNA has three major roles. If you remember those were storing, copying, and transmitting information. Our main focus today is going to be this, copying information, because before a cell divides, it must make a complete copy of every one of its genes. Now we have to recall the components and the structure of DNA. So remember, DNA is a nucleic acid and it's made up of nucleotides joined into a chain by covalent bonds. So remember the sugar phosphate backbone, or again, here is the phosphate, here's the sugar. That's how those are connected in the backbone, and these are covalent bonds. Okay. Nucleotides had three parts: our phosphate group, our deoxyribose sugar, and our nitrogenous base. And again, the base will vary between adenine, guanine, cytosine, or thymine. Okay, those were our four nitrogenous bases for DNA. Now, again, the way that we label the sugar, okay, is based on the carbon number, and this is going to be important when we talk about the direction of DNA, okay? But covalent bonds between nucleotides here, this is what makes up the sugar phosphate backbone. Now, our double helix model, okay, we know DNA has two strands, and if you recall, they are anti-parallel, meaning that they run in opposite directions. So that's where that 3', 5' comes in. If one strand is going up, the other strand will be going in the opposite direction. And 3', 5' just means the direction that it's running. So instead of like north, south, east, west, we just use 3', 5'. The bases, the nitrogenous bases, are held together with hydrogen bonds, okay, here in the middle. And it's important that those are hydrogen bonds because they are relatively strong, but also fairly easy to break, okay, which we have to be able to do in order to replicate our DNA. And then we also remember our base pairing rules, that adenine will only pair with thymine, as seen here, and guanine will only pair with cytosine, as seen here. So last, just kind of reminder on the structure of DNA, quick overview. Here's your nucleotide, your three parts. All right, here's kind of the overall picture of DNA. Again, 3', okay, on one side would be connected to the 5' end on the other side because they are anti-parallel. Here we have our sugar phosphate backbone, okay, again, covalent bonds here, and then your bases in the middle are connected by hydrogen bonds. So those are, again, just kind of the basic components of DNA for the structure, if you remember that from Unit 2. Now, we talked about the cell cycle in lesson 3.6, but we're going to go ahead and review, okay, the cell cycle. So remember, there were two main phases, interphase and the M phase. Interphase consisted of our G1S and G2. The M phase consists of mitosis and cytokinesis. Now, this is going to be our main focus today, is on this interphase, okay? So G1, G2, both growth phases, but the S phase, that's the stage where DNA is replicated, and that's a vocabulary term, so make sure to get that one down. So new DNA is synthesized when the chromosomes are replicated, and at the end of this stage, the cell is going to contain twice as much DNA as it did in the beginning. So how exactly is DNA replicated? Well, the process is somewhat complicated, but we're going to look at the overview and then we're going to get into some details on it. So before a cell divides, it duplicates its DNA through replication. That ensures that each resulting cell is going to have the same complete set of DNA molecules. So here's going to be our overview. The two strands of the double helix separate, and that's going to allow two replication forks to form. As the new strand is synthesized, new bases are added using base pairing rules. And we remember A pairs with T and G pairs with C. Our result is going to be two DNA molecules that are identical to each other and to the original DNA molecule. So replication is what we call semi-conservative, meaning that each new DNA molecule results from the replication has one original strand and one new strand. So here's kind of a picture of what that semi-conservative means. So the purple is our original strand here. Okay, it's unwound. And then the red strand is the new strand. So your two new strands are going to be what we call semi-conservative, or replication is semi-conservative, because you have one strand from the original and you have one strand from the new DNA molecule. So go ahead and pause here. We reached our first check of understanding. Try to answer this on your own. Okay, so let's go ahead and check yourself. So remember, the nitrogenous base pairs were held together with hydrogen bonds, and A paired with T, C paired with G. That anti-parallel meant, again, DNA runs in opposite directions, and that allows the nitrogenous bases to contact in the middle. Each strand has a 5' end and a 3' end, so if one strand's running 5 to 3, the connecting will be 3 to 5. What were the two main phases of the cell cycle? And that was interphase and M phase. Okay, for the stage that DNA replication occurs, that is in the S phase, which is an interphase. Number 5, when DNA is replicated, each new DNA molecule resulting from replication has one original strand and one new strand. So that's that semi-conservative. And number 6, so when replicating this strand, this is the first time you've seen that 3', 5', basically you just flip it. So 3' connects to a 5', A goes with T, G goes with C, C goes with G, and so on all the way till the end. And where you have your 5', that's now going to be a 3'. Okay, again, because that anti-parallel concept of DNA. So if you see something like this where it says write out the complementary strand, that's all that you are doing is matching up the base pairs and the ends. Okay. So here again is continuing with the overview. This is a really good diagram kind of what's happening. So your result is two DNA molecules that are identical to each other and to the original. Again, it's semi-conservative, and cells need to quickly copy their DNA with very few errors. And to do so, they use a variety of enzymes and proteins which work together to make sure DNA replication is performed accurately. Now, which enzymes are important, or what are the main enzymes involved in replication? So there are many, okay? There are two main ones that you need to know, but beyond this, there are so many not listed here. So DNA helicase unzips a molecule of DNA by breaking hydrogen bonds between the base pairs, whereas DNA polymerase joins nucleotides to synthesize a new complementary strand of DNA and proofreads the strand to ensure accuracy in replication. So go ahead and kind of pause here and just examine this photo for a minute. Okay, so we're going to look at this in more detail. So again, there are many enzymes, but right here are the two that you need to know. Down here, this is additional information because we want you to make sure that you understand there are many other things going on. Now, helicase is shown here, okay? And again, that's unzipping that DNA, okay? The polymerase shown here and here, okay? Both of those are DNA polymerase. As you can see, those are joining the new nucleotides, okay, to the complementary strand, and it's also proofreading as it does that. Now getting into a little bit more detail. So DNA polymerase, now shown in this diagram, it's kind of this white clear circle, okay? It can only synthesize DNA in the 5 to 3 direction. So we can see that polymerase moving this way, 5 to 3. So remember, because a DNA is double helix, it's always anti-parallel. So one strand's going to run 5 to 3, other one's going to run 3 to 5. So we call these two strands two different things. We have a leading strand and a lagging strand. So the DNA strand that's replicated continuously without breaks in the 5 to 3 direction is called our leading strand here. It is continuous because it's moving towards the replication fork, and the replication fork is just the area in which helicase has unzipped and the area in which it's moving towards. The lagging strand, okay, is on this side, all right? It's the DNA strand that's replicated discontinuously or kind of in like pieces or fragments. It's also replicated in the 5 to 3 direction, but it's discontinuous because it's moving away from the replication fork. Okay, so as it does that, the polymerase has to come off, and then whenever it does that, it forms these fragments. So here's a fragment, here's a fragment. And we call these Okazaki fragments. And again, those are fragments of DNA replicated on the lagging strand. So Okazaki fragments are only going to be found on the lagging strand. Okay, they will not be found on the leading strand. So here again is kind of a good overview. All right, kind of pause here, just examine this photo for a moment. Okay, So here's the replication fork. Okay, that's the area or origin where helicase, which is one of our enzymes, breaks apart those hydrogen bonds between the base pairs and the DNA can begin to be replicated. Then we have your DNA polymerase, which is shown here kind of as this yellow piece. So there's two there. Okay, and your polymerase on both strands are adding in new nucleotides and proofreading. The leading strand, shown here, okay, is replicated continuously and it's moving towards the replication fork. The lagging strand, shown here, is not replicated continuously. It's moving away from the replication fork. And on the lagging strand, we have these Okazaki fragments, okay, not shown too great here, but only the lagging strand has Okazaki fragments, and those again form as a result of that lagging strand not being continuously replicated. Okay, so now we're going to talk about chromosomes and how DNA is packaged. So even a small cell like the bacterium E. coli has a ton of genetic information. So the total length of the bacterium's DNA molecule is 1.6 millimeters, which is 1,000 times longer than the cell itself. And cells can only handle such large molecules by careful packaging. So genetic information is bundled into packages that we call chromosomes. So chromosomes are the thread-like structures of DNA and protein that contain genetic information. Now, prokaryotes and eukaryotes, their chromosomes are slightly different. So prokaryotes have usually a single circular DNA chromosome that contains all of the cell's genetic information and it's found in the cytoplasm. So here's a good diagram showing the prokaryotic chromosome. Okay, again, just circular within the cytoplasm. Eukaryotes, genetic information is found in the nucleus. They generally have much more DNA compared to prokaryotes, so they contain multiple chromosomes, and the way that they are packaged is a little bit more complicated. So here, okay, that's that repeat of information, but chromatin is the substance found in eukaryotic chromosomes that consist of DNA tightly coiled around histones. So if we look at this kind of diagram, we have DNA, we have histone proteins. Okay, the DNA wraps tightly around the histone proteins to form a nucleosome. Okay, nucleosomes then form these things called coils. The coils then form these things called supercoils. And then chromatin is basically just supercoiled DNA. Okay, again, chromatin's almost like a bowl of spaghetti. The chromosomes themselves are just condensed chromatin. So it's kind of like a somewhat organized way to package chromatin. A chromatid, if we look here at this X, okay, our typical chromosome, here is a chromatid, or one leg, and here's a chromatid, or one leg. Okay, so one chromosome before replication is usually just one chromatid. But when replicated, we're going to look at something that looks like this. Okay, two sister chromatids, which are now held together by a centromere here in the middle. And chromosomes make it possible to separate DNA precisely during cell division. Okay, so here are the terms we talked about so far. This all together is the chromosome. Okay, the chromosome again is found in the nucleus of eukaryotic cells. Okay, chromosomes are made up of chromatin, which if you look in this diagram, that's again kind of this packaged, tightly supercoiled DNA. The chromatids, okay, here's a chromatid. Again, it says one of the two identical strands, so this is a chromatid, and then this side is a chromatid. So there are two sister chromatids in this one chromosome. The centromere here in the middle is what joins the chromatids together. And then we also have these things called telomeres. And telomeres are the ends of eukaryotic chromosomes. Okay, you can kind of think of these like the hard parts on the tips of your shoelaces, and they help to prevent your laces from fraying. Telomerase is a special enzyme that can replicate the chromosome ends, and it helps to prevent genes being damaged or lost during replication. So there are some slight differences between eukaryotic and prokaryotic DNA replication. So in eukaryotic cells, replication can begin at dozens or even hundreds of places along the molecule. So here we have one molecule of DNA. So as that happens, it's going to proceed in both directions until the chromosome is completely copied. Okay, and we have unreplicated down here, unreplicated. All the forks are forming, again, because we can have that start at many different points. There are many proteins that check the DNA for damage or mismatches, but sometimes damaged regions of DNA are still replicated. That's going to result in sometimes mutations, which we'll talk about in Unit 4. Now, prokaryotic replication is a little bit different. Again, because the prokaryotic DNA is packaged in a single circular chromosome, so you can kind of imagine this being that single circular chromosome, and it's going to usually start from a single point and it's going to proceed in both directions until the entire chromosome is copied. Then, if you remember the cell cycle for prokaryotes, we talked about that last lesson as well. That is in a process of cell division called binary fission. Okay, it was asexual. And here's a good overview. Okay, you have your circular DNA, it duplicates, the cell membrane is going to indent, and then at the end, you have a cell that divides and two new identical daughter cells form. Okay, we have reached our exit ticket. Go ahead and pause here and try this on your own. Okay, number 1, DNA polymerase was responsible for the addition to new nucleotides as well as proofreading. Number 2, DNA helicase was responsible for the separation of the double helix. DNA polymerase synthesized DNA in the 5 to 3 direction. Number 4, the segments of DNA replicated on the lagging strand were called Okazaki fragments. And number 5, prokaryotes had a single circular chromosome, whereas eukaryotes, chromosomes consist of DNA tightly packed together with proteins to form chromatin. So that's the end of lesson 3.7. Thank you for watching. Please feel free to reach out with any questions, and have a good day.